Good afternoon, my dear fellow colleagues, research fellows and students. So today our research track group, AI in Computational Biology and Bioinformatics, has arranged one open discussion session, that is thermodynamics of living systems. So here the panel members, I am introducing panel member. So Professor N. Sukumar, you all knew, yeah, his expertise in physical chemistry and application is AI. So Professor Kelit Murali Manoj, our KMM sir, so his expertise you know, yeah, and he is actually pioneer to introduce in his famous Marvin theory, okay. And Professor A.K. Namdakumar, his specialization is material science and he is in Department of Physics. And Professor Ajit K. Sharma, he is HOD in Department of Physics, IIT Jammu. And Professor Sai Sundar Krishna, you know, yeah, his expertise in AI in biology. Okay, so sir, I am just handing over you the session and please conduct this session. Hope you will enjoy this session too much.
Okay. So to introduce the topic, so we've all been told that thermodynamics is a theory of experience. It's not a kind of a fundamental first principles kind of theory, but it's based on in our experience. Nevertheless, thermodynamics is now applied to areas which are actually far removed from our direct experiences. We have thermodynamics of black holes, for instance. Nobody has actually gone into a black hole and studied it. And some of the other areas that we'll be talking about today in biological systems and all that, the applications of thermodynamics and thermodynamics theories, how far they are valid. I think Harish should come up forwards. Right at the back. Yeah, we moved everybody forwards. So, please. Please come forward. Just come forward. 2 rows and all that, so that I don't have to shout. Okay. So what we will be so for instance it has been said that some of the like the first law of thermodynamics, these are actually they are precepts because they are purely held precepts, and that is because scientists want it to be so, and we actually modify our definitions in order to make the first law of thermodynamics come out right. That's a topic for another discussion. I'll not dwell on that. But if you look at Lewis and Randall's book on chemical thermodynamics, in the preface, they compare thermodynamics to a grand European cathedral. And it's this complete structure which looks very grand from the outside, and it looks like a picture of perfection. Okay, so that's so that's what they compare thermodynamics is. So it's like a complete structure. There's not it's done. It's old. There's nothing that you can doing research in. But then when you go inside, he says that you go to the back area behind the shrine and all that, and then you'll see scaffolding is there, some construction is going on and things like that, which is not visible from the outside. Okay, so this is how he compares thermodynamics to. That means for when we teach it in school and in the undergraduate levels, we teach it as a complete theory, as a done theory. But there are areas which are under construction, shall we say. So those are some of the areas that we'll be exploring. So my first question would be to Professor Ajit Sharma. What do you see are the main challenges when you have worked on biomolecules, on ribosome kinetics. When you are applying thermodynamics to biological systems, to the cell, how far are concepts like non-equilibrium thermodynamics valid? Are these all close to equilibrium systems, or are there systems far from equilibrium where we need to take things further? And what are the challenges that you see?
First of all, I just want to say something about thermodynamics itself. So, for example, if you...
just
Yeah.
1 second. I forgot to tell you a little bit the format. So what we will be, I've already shared the format with the panelists. So I'll ask a question, and then each panelist will have 10 minutes to answer, and then I'll go to the next panelist, and again we'll have 10 minutes to answer. And as we said, that there's another program here in this hall at 4 o'clock, so they will throw us out at 3:45. So I request all panelists to please stick to that time, and I will give a 1 minute warning before that. And in the last half hour, and then so that will be the first round. Second round will be follow-up. You can follow up on things that other panelists have said, or if you have something to add, you can follow up, and that will be for 5 minutes each. And then finally, last half hour we'll open it up to the audience for your questions and your feedback and any suggestions. So, sorry about that.
Yeah. So I just want to say something about thermodynamics as a topic itself. As a physicist, or scientist in general, they say ultimately everything you see, ultimately it boils down to physics and chemistry. So I slightly modified it. Ultimately everything boils down to physics and chemistry. But if you boil it more, it's always the physics. So everything is governed by the laws of physics, but you need to have that drishti to appreciate this, to see it from that point of view. And for example, I was talking to Bishwajit today, and we were talking about biological systems. In biological systems, A molecule influence B. Something happens. Whenever chemical concentration of something increases, decreases, right? And ultimately, you can think of it as a point of view of information processing, right? So in biological reaction networks, things happen. Ultimately, it is information processing. So we need to have that point of view to look at things from that lens. And similarly, thermodynamics is also universal, right? So everything, you have transfer of heat, information processing also has overlap with thermodynamics, which has been very nicely described by Landauer, right? Landauer, yeah. So every system, just like biological system, there is a thermodynamics in it, and it is the thermodynamics that governs the direction of any process. Basically, the second law of thermodynamics, right? So thermodynamics is everywhere. You just have that vision or drishti to look at it from that point of view. Now coming to Sir's question about the areas, right, where we are lacking. For example, which are the areas where we can have more push. So thermodynamics, the equilibrium thermodynamics is there from long time, right? And then we have stochastic thermodynamics, which has been, at its peak around 90s, where we had fluctuation theorems, different kind of, which is basically the generalization of second law of thermodynamics, more, and that's how the field has evolved. So, as Vishwajit was saying in the morning, that the field is totally, driven by mathematicians, who they know what is happening there, but in terms of implications of those mathematical principles which have been explored, I think we are lacking somewhere. So for example, we know the second law of thermodynamics. We know that it governs which way the process will evolve, right? It applies some constraints on a process, more specifically biological processes as well. Similarly, fluctuation relations, which are the generalization of second law of thermodynamics, it also applies a constraint, but the constraints here are as I said, second law of thermodynamics is a simplified version of fluctuation theorem, but we don't know in the context of biology, how these constraints can be applied. What are the natural constraints applied by physics, right? So perhaps to me, I mean, understanding how these mathematical principles, which we understand now, plays out in the context of biology or other stochastic processes. So this is one of the areas I think which is very promising. Another one is information thermodynamics, information theoretic approach. So Landauer said that I hope some of you are familiar with the principle of the concept of Maxwell's demon. So there is a demon, so this was it remained an open question that I don't want to talk about the Maxwell's demon here, but ultimately when the debate happened, what turns out is that information is a physical quantity, and whenever you erase information, you need to dissipate some energy, right? And it has a physical consequences in biological system, because as I said in the beginning, ultimately anything that happens is information processing, and it requires some thermodynamic operation. So this is something which I think is very promising, and still we need to explore those two areas. This is what the point of view I have.
Let's turn to Professor Nandakumar, and I ask you, continuing on that, so you work on surface physics and materials, nanomaterials. So when you are talking about surface phenomena, physics in confined media like cells and all that, how valid are the principles that we apply, and what is it that you have seen as an experimentalist which leads you to say that okay, this I can do, but this is something that we need to be careful.
Well, first thing, the focal theme of the panel discussion, which is on animate living organisms, I should right away declare that I have not worked on any animate things. Okay, all my studies have been on inanimate things. In fact, the first preface to any book on material science, Callister, for example, would be we are not going to deal with wood, plants, right, animals, cell tissues. Okay. So we have a very clear definition of what really comes within the code of material science, right? And that is, you could have simply say it's maybe metals or alloys and semiconductors or insulators. So these are the stuff that we work on. But if you strip down all these science and look at the mathematics, you would find many parallel things happening with biological systems. Right? You would find it in economics too. Okay? So what is permeating throughout, I believe, is the common mathematics that tracks the evolution of the system, whether it is a material inanimate system that is evolving naturally under natural driving forces, or any other animate system that is also evolving naturally. Aging, for example. A material can age, so can humans. Okay? So they are all sort of governed with inflation, for example, in economics. So these are things that have some common, some element of mathematics that is quite common to them. My point was that I work on inanimate things, and as Dr. Sukumar asked me, have we encountered sort of exotic systems, confined systems that seems to show some behavior that don't fit into traditional theories. Okay, And to answer that, I should tell you right away that I teach thermodynamics to the undergraduate students, and most of my time goes away in defining equilibrium in the first place to the students. Okay. How do you define the concept of equilibrium? And almost 90% of the course, including thermal physics and statistical mechanics, is on equilibrium systems. I believe Ajit sir would also agree with me. He also teaches statistical mechanics at IIT Jammu. So most of our time goes away in defining equilibrium in different ways. Right? For example, you take the best example I could give you is take a container, adiabatically insulated, ensure that it has a partition. Right? One part is filled with gas, the other part is vacuum, right? Completely evacuated. And so this system on the left has some energy, some internal energy, some entropy, some temperature, some volume, specific volume, and so on. The other part is completely empty. Now what you do is you prick this membrane partition and permit this to leak to the other side. Now you wait for some time, and then you ask, what is the entropy change? So earlier it was on one side, now it has permeated throughout. You know, what is the entropy change? Okay. Entropy, I believe you have some idea of what is entropy, the measure of disorder or something. Right? Now, in the classical sense, you define entropy as the heat transfer over the temperature at which the transfer happens. Right? Now, this is adiabatically insulated, and therefore, there is no entropy created. Delta Q over T, because there is no delta Q, because it was adiabatically insulated, there is no entropy created at all. Right? However, this is counter to what you would expect in the proper intuitive definition of entropy, which is some disorder has been created, some irreversibility has been captured. Right? Because you know it's never going back to the other half again. Right? Even if you put the partition back, you will not get back the original configuration, that all of them spontaneously go back to the left hand side or to the one side of the partition. And therefore, most of the times what we do is, to calculate the entropy, what we do is we imagine some type of an equilibrium path. Right? Assume that this transition from the left end to the full occupation of the full volume happened through some isothermal path, right? Or some equilibrium path. Right? And fortunately, this entropy being a state function, right? That's the point there. You could calculate the equivalent entropy that would have been created if the container was not adiabatically enclosed. We imagine, we hypothesize equilibrium trajectories. Right? In principle, you never encounter fully equilibrium scenarios, right? Unless you wait for a long time. Right? And in fact, that happens most of the times. Okay? Not only in confined systems. Right? I can give you a hell number of examples. Even our PhD students give us results which completely are counterintuitive to equilibrium. Right? So first thing I ask is, does this obey the Gibbs phase rule? Right? That's a rule of equilibrium. Right? You could have only these many in mutual equilibrium, these many phases in mutual equilibrium. A good many students in their initial years of PhD, they show up a lot of results which are out of equilibrium results. They are not confined systems. Right? The problem here is that you have not permitted time. Right? Time is a clear factor there. That some systems are very sluggish. You need to wait until equilibrium is reached, and then you use your equations. Right? As I said, almost all the things that they are studying, students are studying, are basically on equilibrium stuff. Right? So we have a clear set of equations for equilibrium scenarios. Right? They have certain constraints. As I was saying, there are constraints for that. Right? You need to minimize some amount of energy. You may need to maximize the entropy. So it finally boils down to optimizing or maximizing or minimizing certain terms, certain thermodynamic potentials. Right? So that's the way we usually organize the problem. Right? Is it a problem of you want to find the equilibrium state, you maximize your entropy, right? You minimize your internal energy, right? Or it's a trade-off, right, between two terms, so it becomes a free energy. Okay. Now, this is the situation that was happening until 1950s. Right? Equilibrium thermodynamics was very well established. People had no reason to violate it, and every time an observation was made, it was quite sure that it was obeyed. Right? Until some people started reporting some types of unusual effects. Right? For example, you take a system that is having components A and B, right? And you heat it. You would expect that both of them, all A and B, would mix up randomly, right? But people found that in certain systems, like for example gold and cadmium, for AUCD system, for example, you would find that the AU or type A would arrange systematically, right? So every 10 or 20 nanometers, there is AU, and then you have a disordered region of mixture of AU and CD, for example, and then you will have again AU, right? So there was a composition wave that was traveling. Right? So this was not really explained with the theory of equilibrium thermodynamics, right? So therein people realized that we need alternate equations, right? Alternate equations. What evolved was multiple driving forces acting on the system. So we had the Onsager's relation, which considers fluxes, right? So equilibrium can be in two ways. One is static equilibrium, the other one is dynamic equilibrium. Dynamic equilibrium means you take a body, an incandescent lamp, that you switch on the light, it starts glowing and it radiates heat. Right? Now you keep another body beside it, this will gather that heat. Right? And eventually both of them will reach one steady state temperature. Right? This guy reaches a certain temperature, the other guy also reaches a steady state temperature. Now what is actually happening there is, because both the bodies are above 0 Kelvin, they are radiating. Right? One guy radiates, the other guy also radiates. Right? But eventually they achieve a steady state or equilibrium temperature. Right? This is because of a dynamic balance. Right? Some energy goes out from the bulb, it also takes in some energy. Right? So finally this net flux becomes 0. Right? That was the second definition of equilibrium. Right? Net flux is 0. Net flux of what? It could be mass, it could be energy. Right? So both of these developed into two separate streams. Right? One dealing with the flux, describing equilibrium in terms of flux, the other one describing equilibrium in terms of thermodynamic potentials. Right? So both of them were developing until people realized that you need to move out of equilibrium, at least to some extent deviated from equilibrium, to explain what are called multi-phase mixtures. Right? All these, most of the automobiles you use, they are yeah, thank you. Okay, so therein developed what are called phase field models. Right? So as I said, every now and then we observe systems like this in which you have phase segregation. Right? Technically you are expected to see that everything is randomly mixed in a AB alloy, but you would see that there are regions where there is a segregation. Right? How do you explain that? Right? So we need to move out of equilibrium, at least linearly away from equilibrium. Right? To explain the interface between two systems that are in mutual equilibrium. Right? So this guy is in equilibrium, this guy is in also equilibrium. You keep them together, one of them should dominate. But it seems that both of them are in equilibrium. Okay? So globally some order is preserved or equilibrium is preserved, but locally there might be some misfits here and there. But overall they coexist for the simple reason that the overall system must be in global equilibrium. So even starting from simple phase mixtures, which you would need, for example, the phase field model or something else to explain coexistence of two phases. There are other types of exotic transitions like topological insulators, right? Conducting on the surface, insulating inside, right? Superconducting surfaces, right? Ferromagnetic on the outside, and diamagnetic on the inside. Right? So these are types of things that are sort of, how do you call it, exotic phase transitions or exotic systems. So I think there are a lot more problems. I don't know how they apply to biological systems.
Okay, they've given us a lot to think about. My next question is to Professor Murli. So you have spent a lot of your career challenging the ideas of energy production in biological systems and ion transport in biological systems. What do you see as the open questions in the field of thermodynamics of biological systems and bioenergetics that need to be addressed?
Thank you, Professor Sukumar. Basically, I'm a biologist. Like Ajit said, we are at the lower rungs of the hierarchy of sciences. I'm basically interested in nature, and I started working for my postdoctoral. I was an enzymologist, and the problems that I visited in my postdoctoral stint in Urbana-Champaign, it brought me to question several classical understandings. That is, let me give you a bird's eye view of how biological systems were perceived until 10 years back. Mostly, the idea was that things are vitally deterministic. All physical laws somehow lost their context when it came to explaining biological phenomena. Biologists would say that the protein changes conformation, and that would be something that would be adequate to overcome the thermodynamic barriers and all that. Of course, we have non-equilibrium conditions and all that applying even beyond that. Very simple ideas. For example, when I was studying in college level, certain things that were taught looked apparently outlandish, too much of a stretch of an imagination, if I might say. There were multiple Nobel recognized ideas, and I thought they were not right. But then, when I was in Urbana-Champaign, I just hit upon some enzymes that worked in rather unique ways. That is, their activities were very non-specific, and the reaction stoichiometry was rather variable, not like classical reactions. So here is the idea that we posed with respect to the classical biological paradigm. The classical biological paradigm considered the cell to be covered by a membrane which was somewhat intelligent, so as to say, and the activities of the cells were governed by enzymes. Catalysis and the overall outcomes were governed by enzymes, which were defined by the sequence that the DNA gives. So the central dogma governed everything, and the protein would interact with a small molecule at an active site. So, and then these were all subjected to, of course, the quantum chemistry processes for explaining at a molecular level, and at the macroscopic level, classical thermodynamic treatments. So I saw that many times the fundamental concept of having a active site, the transition state based simple definition of very high affinity bound intermediate, like what you were talking about the other day in DNA transcribing or translation mechanisms, not good for explaining the most fundamental reactions. The most fundamental reactions that are responsible for energy production, let's say respiration, photosynthesis, thermoregulations and all that, whether ion fluxes, all that. So what it brought me to is challenging a lot of kind of ideas that enzymes are freely reversible. All reactions can go this way, that way. For example, ATP production. ADP and phosphate is supposed to give ATP, and ATP and water give that. So it's a freely reversible reaction. Lactate dehydrogenase, for example, some of the enzymes. So I would not go into too much technicalities, but for understanding or explaining the simplicity of it, I would say some simple systems like cyanobacteria. Cyanobacteria has both photosynthesis and respiratory system, something that moves electrons from NADH to oxygen to make water, and then it also has, from water the electrons should be moved to NADPH or NADH to make the other way around. So they are all located on the same membrane. Somehow it doesn't make thermodynamic or electrical or electrochemical sense how this could be achieved by the when there are one intermediate common to them. So how much time do I have? I'm midway through. So this is one, cyanobacteria. And very importantly, the Nobel recognized idea of Peter Mitchell for explaining the overall energy production is based on proton pumping. Something which, if you calculate based on simple equilibrium, the amount of water in a cyanobacteria cannot give any free protons, because dissociation of water will not give that proton. So simple ideas like this were quite standing out. So when I reached out to the people who were responsible for these ideas, they would not defend it. They could not defend it. So I thought that this calls for going beyond the ideas that are prevalent, and application of an outside the active site kind of an idea, which could bring in more stochastic phenomena that could support and explain classical biological phenomena. That is, not just saying that, oh, that protein changes conformation as a magical step that could explain. Rather, now you have a diffusible reactive species which could have the the potential to explain reaction. So in my viewpoint, the classical perception of seeing living beings as absolutely vitally deterministic systems needs to be broken, and the Murborn concept which I have proposed is that there are so many ways in which the living beings generate effective charge separation mechanisms, which is why we need oxygen. Currently, what we believe is that oxygen stays bound to a protein in mitochondria to make water. What I'm saying is oxygen is needed to make diffusible reactive species like negatively charged superoxide, hydroxyl radical, high reaction potential species, which can give catalysis, phase changes, electrical activities, and thermal activities, mechanical activities, all that. So this is a complete, perhaps the largest, biggest paradigm shift in biological systems that I believe has happened in any time, even when you compare to genes. So what this does is, it changes the complete scenario with respect to understanding molecular level phenomena as well as macroscopic phenomena. Thermodynamics comes at a macroscopic level. So now if you wanted to compute the, as per the textbook, if you say photosynthesis or respiration and try to gauge at each level, in spite of there being elaborate reactions in the textbooks, they would not be computing. They would violate the first law. They clearly, the second law would also be violated. So, in spite of such blaring problems existing in the textbooks, which I pointed out, the community continued to be sticking on. Now, we have come to a juncture where we can explain from basic ideas the fundamental process like respiration. The most fundamental process of life is respiration, because it is what gives us powering. Powering, any living system that does useful work needs to have a powering module. So respiration is what is that. So what I say is oxygen's role, which makes us intelligent also, is to make that effective charge separation, giving superoxide, and that is in standard free energy change is minus 250 kilojoules per mole. And that's the most fundamental thing that we need oxygen for, and that has been overlooked. So my approach towards understanding life can give you very tangible thermodynamics, kinetics based explanations for why cyanide can kill at such small doses based on basic already known equations of thermodynamics. Even if people say that is not applicable and all that, we can clearly understand fundamental phenomena, why structures are like this. And even if with thermodynamics, we can also extend with it to kinetics, because Marcus theory has shown that thermodynamics and kinetics are connected, and only for some cases like spin rules and all that, it really is not extendable. So generally, what I want to say is this new, one more minute. So this new perspective, that is to think beyond active sites, to think beyond central dogma, to think of diffusible species mediating chaos-ridden stochastic process in life as a contributing element that supports life. For the most fundamental bioenergetic paradigm, currently we believe that ions are pumped out and then they come back. There is no way. Basically, enzymes can only bring things to equilibrium. That definition that some membrane embedded enzyme can just pump things at will is just extending too much vitalistic determinism into scheme of things, not really abiding by the laws of physics. So what I'm saying is, the new Murborn perspective gives us a way to understand biology, keeping intact our ideas on thermodynamics and the fundamental laws of physics that we have studied in school without the need to violate it. So for the first time, we have a tangible set of ideas to start understanding life with, based on the basic laws of physics that we can derive from Noether's theorem and laws of thermodynamics. So with that, I think I'm almost, thank you so much for the time. Thank you for the time. Yeah.
Okay, thank you. My next question is for Professor SAI. So you have a wide range of experience in both academia and industry, biological systems, computational systems. So going forward, where do you see the role of AI in elucidating some of the challenges in the application of thermodynamics to biological systems and some of the problems that we have been talking about?
Great, thank you, Professor Sugmarsh. So let me take a step back. I'll also play to the gallery a little bit, because let's look at the broader perspective first. We've already come across machine learning systems. If you think really about what is machine learning, a machine is some device which does useful work, right? And what is the useful work done here by the machine? It learns. And how does it learn? It learns by maximizing the likelihood. You're giving the machine a data, it maximizes the likelihood, and it learns its parameters, right? So the other way of looking at it, like we are already into the thermodynamic concept of entropy. The moment you say maximizing the likelihood, you are minimizing the negative log likelihood. And that's how a loss function is really written. And this is exactly what is entropy minimization. So you're looking at places where your knowledge is more certain, right? In a sense, the whole concept of deriving this architecture and going towards likelihood maximization or entropy minimization, you're already in the entropy terms. You're already mathematically put one leg into entropy. Now, one more leg, you think about Gibbs free energy. That's the second most important fundamental thing, right? We have learned in our physics undergrad, delta G is equal to delta H minus T into delta S. So basically, enthalpy minus your temperature scaled entropy is going to give you a Gibbs free energy. Why is this useful? Of course, apart from saying that if it's less than 0, it's spontaneous process, greater than 0, it's a non-spontaneous process, you have to give work for it, and equal to 0 is some kind of reversible process. All this conceptually fine. But if you really think about where this is applied in a real world biological system, think about a cancer propagation. That's actually a thermodynamic process. And that's a huge thing nowadays. Like cancer generally happens, there's in a cancer metastasis, if you look at it, there are a lot of tumor microenvironments. They have different kinds of cells in the tumor, and every cell has its own pH, its own temperature, its own enthalpy, its own entropy and conditions, right? And if you actually overlay the RNA transcriptome, that is the genes expressed in these cancer cells, along their PPI, the protein-protein interactions, Now again, what is the conceptually, how do you think? There are two proteins. There's one protein, I'm standing here. There are only two links that I can go to, A or B. Then it means I'm fairly moving towards a certain entropy state from there on. But if you've got 20 different things, my measure of disorderliness could be very high, right? So think about that being overlaid along with my transcriptome data. And now this, recently researchers are saying that this is very important for looking at the 5-year rate of survival. So that's a kind of open challenge right now. It's kind of coming up. And there are some papers which are already appearing in this domain. In fact, I've seen a paper which basically lists down different kinds of cancer, like glioblastoma, right? What do they call it? GBM, glioblastoma metaform. It's one of the toughest, severe kind of cancers. So it's only 2 or 3% 5-year rate survival. So if you look at the Gibbs free energy, it's very high. So it's kind of what kind of Gibbs free energy you calculated out of your entire tumor microenvironment. It's very important. So similarly, you've got other cancers. What's the next in line? Maybe a small cell lung cancer. The next one could be PDAC. So there is a gradation. There's a gradation in terms of how this Gibbs free energy converts into a 5-year rate of survival. This is very important. The other thing that practically how this is useful is also think about this, instead of considering as a one thermodynamic system, if you look at a lot of microbiothermodynamic systems, which are microbes, microbiomes in you. These microbes have different kinds of biological phenomena applied to them. They grow in a certain way, and there is a co-evolutionary process. There is a competition. There is a commensal pattern, there is a descensal pattern, there is a diversity pattern which they kind of leverage. So all this involve you can actually put this into a thermodynamic framework. Practically there are so many implications of considering this as a very strong complex architecture, and which is being considered nowadays. So this is yet another way in which AI and machine learning are using multi-omic principles in looking at these microbiothermodynamic systems. So it's again, in a way, there's a lot of causal reasoning and machine learning involved in characterizing all these systems, which can be put into this thermodynamic framework, and you can also think about how this is going to progress in the future. For example, we are working on a research which looks at this longitudinal microbiomics. So you look at a person, you track him over time. His gut actually has thousands of bacteria, and these bacteria has certain alpha diversity. This pattern keeps changing over time. So depending on how much fiber he takes in, that amount of fiber is actually, your body cannot digest fiber. That may be shock to some of you. Body really cannot digest fiber. Your fiber is basically for the gut microbes. They actually eat those fiber and release something called short chain fatty acids. And this short chain fatty acids actually enrich the whole ecosystem. Maybe that will be useful for some lactobacillus and few other bacteria that will get into a commensal pattern, and that will be very useful for your eubiotic state. You don't want your bacteria to get into a dysbiotic state, because if you get into a dysbiotic state, what you do is you eat your gut epithelium, and slowly these cell walls come out. So that is your lipopolysaccharide. That actually enters your LDL, low density lipoprotein. That actually forms these foam cells. These foam cells go and deposit in your artery, and voila, you got a heart attack, cardiac problems, cardiovascular diseases. The similar is the nature for other kinds of insulin resistance, metabolically assisted fatty liver disease, type 2 diabetes, you name it, CKD. I can give you a lot of processes which are actually involving all these kind of micro biothermodynamic systems. So this is very important to actually understand the overall enthalpy, entropy, and the kind of the entropy, the path in which a process is progressing biologically. That's very important. We still have time? Yeah. And just maybe one more example. A lot of people don't know that this whole microbial dynamics, because of this growth, evaluation, descensal, commensal, all these patterns, this actually happens even way before our birth. There are a lot of these placental microbiomes. People think that only during the vaginal delivery versus non-vaginal delivery there's a change in microbiome. Microbiomes kind of accrue over time. That's not true, because nowadays people have found out that in the placenta there are some microbiomes which are already available, which are actually, the fetus actually intakes this microbiome when it's in the womb of the mother. So that actually is shown in the meconium. So they also look at the placental microbiome, the cord blood cell microbiome, and that's again an interesting area in which you can think about. These are a few things that I'm happy to discuss as we go along.
Yeah, thank you. Again, you have given us a lot of food for thought. So now I'd like to open it up to the panel. So again, let's go in the same order and ask you to have any feedback or any follow-up on some of the points that have been raised, or any additional points that you think would be interesting there. That's it.
I think I said what I wanted to say, that if we have that vision to look things from the perspective of thermodynamics, right? Every problem is in some sense a problem related to thermodynamics, yeah, right? And in biological context, which are more stochastic in nature, which are more unpredictable, than the other macroscopic systems, right, which are more deterministic in nature, then the role of thermodynamics, and more specifically stochastic thermodynamics, becomes more relevant, right? And I don't need to name the processes, where it can be applicable. Whenever, as I said, some stochasticity is there, still, biological processes are noisy, stochastic in nature, but still they are good at, performing the design task, making decisions, right? And, sometimes we overlook many things. For example, I'll give you a very simple example. The DNA replication that we have, it's highly accurate. So only, the chances of error are 1 in 10 to the power 9, 1 in billion. How do we get such high accuracy? Thermodynamics is responsible for that.
Is that after the error correction or before the error correction?
So error correction, for example, in DNA replication, you add incorrect nucleotide, and then it goes back, right? So the reversible activity, which is we call it exonuclease activity, when it cleaves the incorrect nucleotide, that it also thermodynamically driven, right? And in, for example, protein synthesis itself, right, it selects the correct amino acid based on the simple thermodynamics, right? Same principles are applicable there as well. In DNA replication, first it initially selects or rejects the nucleotide based on the base pair matching, and then also it brings, if it is incorrect, the exonuclease activity, then it brings some conformational changes to the overall complex, which is again thermodynamically driven. One path is more favorable than others, that's it, right? So ultimately, molecular level, if you go, you choose any biological process, thermodynamics play a critical role. Not only non-equilibrium thermodynamics, which consumes energy, but also equilibrium thermodynamics, when protein folds in a specific three-dimensional structure. Of course, it's a mixed process. Active, it's a result of active and passive processes, but passive processes, right, which simply the process of minimization of free energy is playing an active role there, and then your genes are turned on and turned off, right, which is again a thermodynamic process. You consume ATP molecules, burn some fuel, which is a non-equilibrium process. So everywhere in decision making, energy expenditure is there, entropy production is there, you can relate it to thermodynamics and have a deeper insight into that biological system. Yeah.
Not specifically though, but the scales that you are talking about, at the molecular level, disorder and so on, I believe there are, it's just a point I wanted to add in, if it would help. I believe there are tools now that help us to visualize these types of propagation of stochastic events. Highly accurate, magnified images can be produced of such small structures in situ. So propagation of cancer cells, or it's like looking at deformation in an alloy. Right? Yes.
What kind of techniques?
There are microscopic techniques. I am not talking about spectroscopic techniques. Right? Those were existent. They are pretty much common. But nowadays, I believe there have been a lot of reports where live experiments on biological systems are being visualized. I also remember reading a recent paper on a very interesting thing, what is called an indentation. An indentation is something that a mechanical engineer would know. Right? You take a metal plate and bump an indenter on it, a sharp pointer on it. Right? So it, depending on the hardness of that metal, you create a dent. So you
guys probably are thinking in terms of Python.
Oh, okay, I see. Okay, so that's a mechanical procedure, right? So you have a scale, diamond is very hard. So what happens if you put 100 kilogram load on it? It creates a certain indent. So relative to that, you have a different hardness and softness of things. I recently saw a paper in which they carried out this experiment on a cell, biological cell. One that was infected with cancer, and the other one that was a neat cell, a clear cell. Right? So the point of these experiments are that these can be done automatically on thousands of such regions. Right? And they create a statistical measure of the hardness of, the hardness is a macroscopic parameter, right, of a system that is behaving in a stochastic manner, or something that is evolving biologically. Right? So I believe these are at the crossroads of clear interdisciplinary type of thing, where an engineer works alongside a biologist. So I believe these are areas where students, if they are interested, they can probe all these things.
So let me take it away.
Yeah. Well, thank you. No specific questions? Okay, that's better. As Ajit said about information processing requiring energy, and he again followed it through with stochasticity, and Nandakumar also said that. I would like to say that the new perspective that is brought out in Murburn is something that incorporates all these elements. Classically, biology was infused with a high level of determinism, because primarily genes are so deterministic, the errors that you pointed out, very little. So the idea that life has to be very, very, very streamlined into a number of definitive steps, going through clear-cut active sites, were very dominant in our understanding of life. The now emerging idea of Murburn, that incorporates the randomness that molecules and their interactions fundamentally entail. With Murburn, molecule, unbound ions and radicals, stochastically generated. That's the etiology of the word. So with that, we have a complete, while central dogma gave that active site based reactions, Murburn is a set of all reactions that are not necessarily involving that. So now, that is a fundamental contributor to life that was completely oversighted earlier. For example, if let's say oxygen comes, takes an electron, makes superoxide. That was seen as a toxic waste product, right? Now, the new perspective says that's why we need oxygen. We don't need oxygen to go and bind and stay at an active site, at one site. So with that, what do we have? We can explain why cyanide is more toxic. Cyanide is so toxic at say milligram per kg dose. So if we have the classical explanation, it says cyanide goes and binds to heme. That's a 1:1 stoichiometry, and that cannot explain the dosage because the heme proteins, they are the toxic load is say 1. I have thousands of more molecules of heme. So even if I take off one away, I'm not getting it. So here we can incorporate thermodynamics explanation because cyanide can catalyze the conversion of superoxide and hydroxyl radical to water without getting consumed. And that reaction is minus 275 kilojoules per mole. Whereas oxygen getting activated to superoxide is minus 250 kilojoules per mole. So you have a more facile reaction. That's why when cyanide is present, you lose intelligence, vision, all life activities within a fraction of a second. These reactions are highly kinetically facile because they are diffusion limited. So now we have a thermodynamic, kinetic, structure function explanation and probabilistic explanation that could justify the basic physicochemical features of a living cell. So for the first time we have a real statistical explanation also for how some, for example, a mitochondrion is just a random mixture of some molecules with some scattered proteins. There are no definite electrical pathways for it to make transport chains. There is no deterministic mandate for it to pump some ions out. There is no way proton, which is 0.00002 angstroms, it cannot be pumped out like that. And they didn't necessarily calculate the hydration energies and all that. So now we have a completely new mandate or a way to see life with a new thermodynamic, kinetic, structure function correlation, probabilistic, evolutionary perspective. So with that, what we can avail is we can understand the cancerous tissue that Sai was talking. We can approach that's called the energy consumption is higher. They might in the cytoplasm. That is Warburg effect. It has been quite for a long time. It has been known. So these kind of activities of oxygen, what happens in cell, what happens in the cytoplasm versus what happens in mitochondria, all these kinds of demarcations we can understand better. We have to move beyond the active site, beyond the strictures of the central dogma. Central dogma is beautifully deterministic, but it works at a longer time scale. And Mormon is highly stochastic, and it works at a very acute time scale. So our intelligence is not necessarily driven by genes. Our intelligence is by these stochastic reactions. Yeah, thank you. So that's what Mormon does. It's a new thermodynamic window to see life per se. Yeah.
Keep it with you, for instance, because I have a follow-up. So a couple of points. So first of all, when I mentioned this size business, the proton, I think to some of the practitioners, because I was confused about the way they were using it. So I was told that, this is just a kind of convenient terminology that they're using. Because I was wondering, is it proton? Because we talk about protons in water, but there are no protons in water, it's H3 plus. Right?
H3O plus.
It's H3O plus.
I don't know.
Right. So they said, no, it is kind of, so it is kind of a more like you were saying, a diffusive process, but you're saying that it is attaching to one amino acid, protonating it, and then another proton. So it's not exactly that same proton which is going, it's a kind of a diffusive process, but probably not in the sense that of the term that you are using it. Okay. Another thing that so you use the term stochastic and deterministic and statistical and all that. So, I mean, there are a few things like in physics we use deterministic, but within say deterministic and then chaotic, but then there is also deterministic chaos, right? And you have now probabilistic in terms of coarse grain, that you do not lack of knowledge, right, of this stuff. Then you have the quantum effects, which we have not talked about here, and probably that will be beyond our scope. The probabilistic in the equations itself, right? So these are some of the things, any comments on?
Yeah, sure. The proton hopping mechanism, the here and there. So the point with that is the idea that you get, as he was saying, the molecular visualization, imagery, and all that one gets to see in the YouTube videos and tutorials that we get to see. It is, if you want to create a gradient, it has to be physically there. Now, if it is a hydronium ion or a Zundel or Eigen ion, there are different levels of hydrations. Now, they all entail different energy levels. Now, these are not being considered. The idea is very conveniently, so the fundamental thing is, okay, even if you are not considering the dimension, let's just say the availability of proton. Mitochondria is so small, it doesn't have a proton. So how can it pump? There are multiple levels, and now when that is pointed out, they are conveniently moving it to sodium pumping. Now, how? Yeah. That's what we were discussing. So the point is, they are just exchanging a set of Greek gods with Roman gods when there is not this ion pumping, which is basically the extrapolation of the electrical ideas that we have. How an artificially intelligent system works is through some electrical activity. We see some electrical activity. We saw that, our cells, erythrocytes, they have much higher levels of potassium inside versus sodium outside. So this was seen as the foundation of intelligence and the disparity of potentials and the drives. That is, electrical activity was supposed to drive. Now the point here is, now if you have a scooter running, it produces sound and smoke. Biologists thought that the sound and smoke is what is running the scooter. So it is not the potential per se that is driving the cells, it is the internal charge separation inside that creates the oxygen-centered negative species. So that is what is giving the transmembrane potential, if you see what I'm saying. So
correlation is not causation.
Exactly. So they thought that there is a transmembrane potential, so this transmembrane potential is what drives the cell. But the point is, there's not the ion was pumped out. So it is not that. Oxygen comes in, makes superoxide, that becomes negative, that is thermodynamically facile, that is kinetically beautiful, everything is good. They did not see the role of oxygen. So that is completely changing the thermodynamic landscape, the necessity of pumping, the availability of proton to pump, everything. At the grassroots level, it changes. 8 nobles have been clearly, quantitatively have shown that are invalid. In terms of assessing, in terms of thermodynamics, kinetics, structure, function, correlations, if one were to be really critical. Thank you. Yeah.
Professor Sai, any final thoughts before we open it up to the audience?
Yeah, just a couple of things on the points made by KMM and Professor Nandakumar. So on the visualization part, the microscopic visualization, I was just reminded when you were talking about this, there is this interface between the lipid droplet which carries the fatty acids to the mitochondria at the wall, and there is also this endoplasmic reticulum. There's a bunch of proteins, so they do this thing called proximity proteomics nowadays. So that is a very beautiful thing, because you said this microscopic visualization. So that basically gives which are the proteins that are responsible for that payload to get in a most efficient way. So I was just reminded of that. So that kind of facilities have come. In fact, last week I was reading this paper. I was really, the figures, the images it was amazing. On the Warburg effect thing, I mean, I would also request the audience to look at just search up somebody called Thomas Seifert. Warburg effect is known, there's nothing new in that. But the concept that a substrate level phosphorylation or fermentation is what is driving some of the cancer cells for the nature completely shifts cancer from a genomic territory to cancer as a metabolic disorder, as a mitochondrial dysfunction related stuff. So this is something which has to be really carefully studied, and this is beautiful. So these are the 2 points. Yeah, they're all there. They're all the kind of the markers that make you more susceptible for that, but that's not necessarily the metastasis or the propagation kind of, that's not, again, these are, increase your risk factor of doing that, but the real mechanism is something different that's driving it. It's kind of metabolically driven. That's this hypothesis.
The thing is that certain modifications are protecting you from cancer, and certain that when they get defective, they actually turn the cell cancerous and things like
Correct.
that. Where is that?
Again, it's just a risk factor. It's just a risk factor. That's actually primarily true even for
If it's
COVID.
a risk factor, it's a risk factor because it is aiding the metabolism or disrupting the metabolism in some way.
Not deterministically. I mean, it may or may not. That's the only problem. Again, it's a big metabolic pathway, as you know. So, one change in a small upregulation or downregulation somewhere changes the concentration effect somewhere. And since there are multiple pathways, that's where we need to go into something like Murburn to actually really think about it very deeply.
The stress earlier was pure. Where is the stress earlier, like we had that discussion earlier, there was purely on genes being the causative.
Yeah.
Now, the point is with Murburn, the diffusible reactive species can affect genes. It can, the hemoglobin, glycation, everything. It is something that is connecting things. It's diffusible species, they can interconnect. That's how you have a network of interactions even at a cellular level. Now, that can, Seifert's idea that he was talking about is one need not always stick to the genetic protein level. There are
Even the genetic effect, it is a systems effect because that is producing some protein which is metabolizing something, right? Because ultimately it is the small molecules which are coming and
yeah. What I'm saying is, we look at the important things that determine life activity only in terms of something binding something and making an effect. So if or modifying something at a protein level.
Okay, I'll actually answer that in a
sure,
little
sure.
bit. So the thing is, if you think about oxidative stress, I mean the explanation why oxidative stress causes this from a metabolic perspective, that Murburn has a better way because of the pathways. The genes cannot explain that. So that's
yeah. So basically, for example, now let me say, in terms of therapy, if say one molecule, say let's take it as 1 milligram, okay, dose. If you give it, it shows an effect. 10 milligram, no effect. The classical theories, thermodynamic binding based theories, are not really adept at explaining this, because the binding based outcome, the more the molecule, the more you should have binding. So, but now with Murburn, what happens is you have a diffusible radical that is stabilized at a dilute concentration, but ineffective at a higher concentration because they collapse together. So now you have concentration based. In this, the intelligence is not in a membrane protein it is not in a membrane protein or in a nucleus or in a brain, but it is an emergent property that is arising due to molecules and ions and all that interacting. So we need to look at it in a delocalized way, not in a very focused way. This has to come for us to understand diseases like cancer, neuronal problems, because ultimately it is the electronic activity. Currently everything is seen as an ionic activity. The energizing or intelligence, all that is ions. Now there is one electron processes that are very crucial, that are chaotic at its core. I might be using the terms like you said, stochastic, chaotic, deterministic, all that a little here and there, but that is with an intent to explain or understand. The whole concept has to be more, opened up, not just with a very stringent eye like what conventionally biologists were looking at. So I think this is also
Just one quick point to add, which is like, there's a lot of implications for precision medicine, the way we look at this whole thermodynamic framework, and this may not be totally the biomedicine as we know, but we have had traditional forms of precision medicine like Ayurveda. It talks a little bit about the heat produced in the system. So your doshas, dhatus, your malas and agnis. So all are responsible. Dosha is kind of your own kind of a phenotype. You can think about it like that. And based on your phenotype, you not only have a quantitative way of dealing with the heat, but also an experiential, qualitative and relative value of dealing with the heat inside your body. That's very, very paramount to that theory actually. Something which is very profound. You can use that.
It's okay. Yeah, I was just saying, speaking of opening it up, it's time to open it up to the audience. Please feel free to ask questions to any of the panel members or one or more. If you have some comments, you can also make them, but please make it brief within 1 minute or so. So, yeah.
Yeah, we can get the mics. Prashanti, any questions?
Maybe I will start.
Yes.
It's okay. I think my voice is too louder. Okay, recording. So like when we are mathematically modeling the thermodynamic equation, like Gibbs free energy equation, or any, like you are working on the protein synthesis, like optimization. So in mathematical equation, we don't consider the water molecule. Do you consider the water molecule? Do you think the any mathematical thermodynamic equation is 100% will able to make a real world, will able to mimic the real world? What is your idea on that?
So thermodynamics, first of all, I don't do thermodynamics. Okay.
So sorry.
Okay. But your question is water molecules, right? Whether if let's say you are considering one protein molecule, maybe I'm trying to understand its folding, right? Which is one of the you can simulate. You can have molecular simulations of protein folding, right? So normally, yes, in those simulations, we don't consider water molecules explicitly, right? But this is how we have thermodynamics. So in thermodynamics we have something called system and we have something called reservoir. So you can consider water molecule as a part of reservoir, and then it's not
Implicitly.
Yes. So the energy exchange that you have, system to the reservoir, this is nothing but it's interaction with the water molecule.
Students need to go, they have exams, so yeah. Nobody?
So I have just one comment for students. If you are interested in understanding biology from the perspective of thermodynamics, there is a very good website called Physical Lenses of the Cell. So it explains all the biological, not all, but many biological processes happening inside a cell through the lenses of physical sciences, mainly thermodynamics. It's a very good website. I often visit that.
Physical lenses of the cell.
Of the cell.
Yeah, right now. Physical lenses of the cell. When somebody says something like that, just from an experience, so you can't get it. Yeah, sure. Yeah, please. Give them. Yeah, you continue.
Okay, to Professor Nanda Kumar, like material, what is the difference between the, like in the dynamic equilibrium? Is it material also have a dynamic equilibrium?
It depends on whether it is under a constant driving force.
Okay.
So if I take a metal plate and it is put under a temperature gradient, right? So now this is at a steady state temperature, the other end is also at a steady state temperature. So now the system has the temperature gradient, and that gradient is not evolving in time.
Okay.
Right, so then we know that it has to have this temperature over at that end and another temperature at this end, and it has an exponentially decaying temperature gradient.
Yeah, now Dr. KMM sir. I have a simple question. Like if two molecules bind with a protein, and one molecule binding energy is low than the other molecules. Low means minus 5 kilocalorie per mole, and another molecule's binding energy is minus 2 kilocalorie per mole.
Okay. No.
So minus 2 kilocalorie per mole is a inhibitor. Is an inhibitor of that protein. Do you think that minus 5 kilocalorie per mole also will be the inhibitor of that protein?
Well. So minus 2 kilocalorie per mole is one inhibitor, another molecule. It depends on, in the Morborn perspective, it can bind here and there, and it depends on where it binds.
No, I am telling both are binding in the binding site. Active site.
That's the see there in the Morborn perspective. We need to go beyond the active site. That's the whole point. Now the interaction realm is not just one place. It's delocalized in or around the protein.
No, there is a allosteric active site is also there.
There is there.
Okay.
Now I'm talking about a redox process. Now what happens is, in the Morborn view of seeing things, there is a protein, there is a interaction scheme. With the diffusible species can get stabilized on the protein, and then there could be a molecule that could be bound anywhere on the surface. So it can happen that this molecule that is bound away from the active site, say for example cytochrome P450s, the enzymes that metabolize the drugs in our liver. So these drugs don't have evolutionary memory like lipids or proteins or carbs, which are metabolized normally by the normal enzymes. Now the murzymes, which are involving the diffusible reactive species, now they can bind here or there and all that. They can activate, they can inhibit, they can, the same,
Is that binding covalent or non-covalent?
Not necessarily covalent. It can be low affinity, it can be high affinity. The point is that
normally when we are talking about inhibitors, we are talking about non-covalent interactions.
yes, there is mechanism based inhibitions
However,
also.
there are a lot of covalent inhibitors which are now, inhibitors drugs which were previously thought to be non-covalent inhibitors are now realized that they are covalent inhibitors. So the ones that you are talking about, would you say they are primarily covalent or primarily non-covalent? And where is the experimental evidence for that?
Wonderful. This is wonderful. Now the point is, earlier inhibitions were kind of seen in. There are classical enzymes. I'm not questioning the mechanism of those classical enzymes, particularly redox enzymes. They can involve diffusible species. Now your question, coming to that -2, -5. A -2 can be a better inhibitor than a -5, or a -5 can be a better inhibitor, and both need not be also. The point is, the inhibition ability can be dependent on a wide outcomes, stochastic interaction
So
schemes.
as you told that drug, if I take a small drug, some nanomolar, if I take that drug is at 5 nanomolar. 1 nanomolar is working but 5 nanomolar is not working.
Yes.
So behind that there is a lot of reason.
Yes.
It's possible.
Yes. So that logic that for the first time.
Only the thermodynamic is behind that, do you think?
No. What I'm saying is, to explain that, we cannot stick to a binding based logic, because this the idiosyncratic or hormetic dose response, this particular event is called hormesis, wherein a dilute dose of a drug is more effective than a stronger dose of a drug. This is not supporting homeopathy principles. So now how do you explain that based on the available ideas of binding or thermodynamics or kinetics or enzymology? That's the question. Now the point is, when a smaller dose, if it is more effective, it cannot be based on binding. It is based on a secondary catalytic activity in its own might. That is, when you have a diffusible species interacting with this dilute.
Yeah, it can be based on anything. Maybe it is a specific to that target. It's a selective binding.
No,
it can be specific on that also.
No, it cannot be, because 5 nanomolar is not effective. So the logic of binding does not hold. 5 nanomolar is more higher than 1 nanomolar. So you cannot say that.
No,
1
it is
nanomolar.
stronger binding. Maybe it is not working.
No, but then the logic of binding does not come, right?
No, logic of binding definitely is not associated with one molecular bioactivity. That's I'm going to tell. The logic of the binding is not equivalent to the bioactivity of a molecule. Am I right?
No, that if you want to say it like that, you need to have a logic to support that. And what I'm saying is, Murbon gives you that logic.
Okay.
Yeah. That's all.
Yeah.
Did you understand that?
Yeah.
Murbon gives you a potential logic. I'm not saying it is the only logic.
So this non-linearity which you're talking about, so we are collaborating on something, chaperone action. So normally what happens is, you don't have one single pathway to control things, right? So you have multiple pathways. So I think in our model what we have seen is, so whenever you have transcription factor, you increase their concentration, and then you should always see more expression, right? This is what the logic is.
But it's not happened.
But it does not happen.
No, it's equilibrated in some
Yeah. So for example, the one model which we are developing, there are 3 different control modules, right? And if you increase the concentration of the transcription factor, it increases. And then if you increase further, the expression level decreases. So, what he said is, things are more complex than, it may have other consequences. I couldn't fully understand what he was trying to say, but, I think that's the idea, right?
Okay, so last question to Shai. Is AI can completely model a thermodynamics of any living system?
That's so yeah. So I'm sure it's pretty complex problem. Thermodynamics itself is a framework. So how you model it, it's more of an art. Actually, there's no one way to exactly say. It's more of calculation. So I mean, it's just one instance of a system can be modeled, but there's no concrete answer to that.
Lot of machine learning tools are now developing. Yeah. So I know some group of Princeton University, they develop Langevin equation. Just you can use as a machine learning tools, and they actually they give the name Dynamo. Okay, and it published in Cell in 2023. And you can actually make mimic of a cell, and you can get the dynamic information, because one of my collaborator, so Audit knows, so they are doing on that process. So another is actually AI one tools is developing to describe the non-equilibrium, far from non-equilibrium process. One machine learning tools. I noted this one paper. This published in, I think, in PRL. Yeah, it's recently come. So I hope in near future, hope some new machine learning tools will come.
Yeah.
And basically I have some faith on Dynamo, this new concept. Same machine learning tool, Langevin equation can be used as a machine learning tool. I think so. Yeah.
I mean, that's one good island. But I cannot exactly say that it will happen or will not happen. What I can say is the path to it.
Yes.
So
yes.
basically, the path will be multi-agent structure now. So there will be different agents looking at different kind of reaction kinematics and dynamics,
Exactly.
and that's how I think the path is. But how far it will lead, it's because it's still not got into any stage.
Exactly.
So I couldn't be very hopeful of that. But
we need some time, but hopefully,
Is it only about the solving equation or it is something? So is it about only
actually, say one cell, we usually use the intercellular reaction. And we want to get some information. We make some mathematical model. But how actually we can make all information through one dynamic equation? All the information you are mimicking the cell information, and that's why actually in abstract mathematics there is one concept. I just forget the mathematical. Okay. No. So that is actually the topological one concept is there actually. Just now I cannot remember. I will tell you. So this concept actually they are trying. So. Topological concept with this with Langevin dynamics simulation. So that actually they can mimic all the information. So we are getting one information from cell, but how you can describe different cellular information? So we can actually describe the all through AI, and that is the main aim of that project. That is called actually you can search in Dynamo. So they just write this paper in 2023, I think, in Cell.
This is regarding the same vertices continuation. So she said 2 kilocalorie and 5 kilocalorie molecules which have high energies, which is having binding to the same targets. So how thermodynamics will help you to identify? There is a computation between these two. So how thermodynamically you can answer that to say that 5 is or 2 is going and binding to the same, or you have any structural modifications to say that?
I don't think at this, with that level of information, just that binding energy value, it would be possible if in a classical enzyme, but not necessarily in a mosaic. You need to see what is the reaction that is involved to be in a situation to predict. Because if it is a simple interaction like high affinity interactions, like what he deals with usually in his translation machinery and all that, then you can make more or less predictions, definite predictions. But in the kind of realm that redox reactions are involved, very difficult to predict. Alright? Yeah.
I will add one thing. Like AlphaFold recently captured the protein folding. The protein folding, as per my knowledge, this is just the thermodynamics. The actually it's happened due to the thermodynamics. So AI captured little, but maybe not all.
It's interactions basically, not the,
basically the atomic interaction somehow is mimic.
It's not following the kinetics actually. But
it's not following the kinetics, but somehow it's mimic at least.
Any final comments? Any questions from the audience? Any final comments from the panel?
So thank you panelists for enlightening us with your knowledge.
Yes. So maybe silly, but let me ask the question. So if we think life as something like a set of events, say for example we have billions of people and we take those processes as events. So in the probabilistic perspective, right, everything is going somewhat, there are slight deviations from the say a deterministic pathway. Right, say if we take something like a cancer, we have say most of the things are going in a particular path. So that may be the reason why people are modeling it as a deterministic system, maybe approximation from the engineer's perspective. So
but otherwise it would look like something like a biased, very very biased system which tends towards a particular process. So how does your theories, to Murli sir, so say for example a very minuscule concentration of something can cause large effect in comparison to say higher dosage, how does that fit into this kind of
Even like things like market forces,
yeah.
they have been modeled by purely deterministic equations, but also through agent-based, stochastic
Yes.
process.
Yes.
Yeah. See, the point is now you have.
With your final.
Yeah, final. So, thank you so much. Thank you so much. I really appreciate this. Now you have say 1 milligram. You have said 10 milligram. You don't get an effect in 10 milligram, but you get effect in 1 milligram. This is your question, right? So now, wonderful. Now, classical theories of outcomes require this molecule to go and bind to something. So if you have more of that, you have more binding. This binding based intermediate is what the 2 calorie energy that they were talking about.
Unless you have saturation.
Yeah, and unless you have saturation. Yeah, of course.
Kinetic this thing, so you have.
But even in saturation case, 10 should give saturation, 2 should also give saturation. So then you cannot explain why I don't get in 10, why I get in 2. So the point, my paper was published in dose responses, the journal, American journal, that was looking for an explanation for this. This hormesis is known to be a reality in biological systems. Alright, it's not something that homeopaths just claim like that. But homeopathy is a little crazy because once it goes to dilution beyond a particular level, there is hardly any molecule left. So the thing that you particularly asked is how can a small number of molecule give a larger effect when a larger number of molecules don't give that effect. So the classical explanations cannot give. The Murborn explanation is now you go beyond this binding based activity. You go to now this small molecule is giving a radical, it's giving a reactive species. Now that reactive species is getting localized at a particular area. And now if there are a lot of those molecules, what happens is they are radicals, they coalesce, they cannot give activity. When they are smaller, they can get stabilized and they can give a secondary catalytic activity. And
Brown ring test.
pardon?
Brown ring test.
Correct. In school qualitative reaction. Yes. So you got that answer?
Yeah.
Okay.
So, but again, what I was asking is like, say if I have these set of events, right? So set of a number of like millions of events leading to a particular macroscopic property, is it like it is going to affect at a very very fundamental step event such that you can correct all these things there? Or
Well, this is a paradigm shift in a very fundamental way. Why? I'll tell you. Now the problem is, now you can take a fruit, you can put it in a mixer, you can get juice and pulp. You cannot take juice and pulp, reverse the direction of mixer, invert the mixer and get the fruit. You have to have a different way to get the fruit. And Murburn is that way. Now you the point is, the point is, you need certain ways to understand. So now you have an energy, let's say, it's not a determinate, you have water being poured over a mountain. It comes down. The landscape is such that it will come down. The drive, there is not enough drive in the chemical reactions that we are seeing in the textbooks. They are all -20, -30 at the most. ATP hydrolysis gives -36 kilojoules per mole. This is the standard thermodynamics treatment. So with the 1 electron reactions, the energy yields is -500, -600. So now you can have radiations produced. You can have one molecule communicating with another molecule that is located distantly. So now you have opened up a kind of a completely unknown interactions and governing principles, operations in understanding biology. So now we have evolved from chaos.
But there will be
Oh, definitely. The point is, until now, all the experimental observations are abiding by Morburn. The classical explanations don't meet the basic requirement of first law and second law. That is the problem. That's the reason. But what happens is, usually people say that, oh, you bring a new experimental evidence. I have shown that until now, whatever were the experiments that were treated as support for the ion pumping and all that, they were purely misinterpreted, purely false ideas. So your question is, how does this change our understanding in the scheme of things, and that has happened or that will come in the future? Now, for example, let me say, two people with genetically identical, will live. I'll just sum it. Two people with identical genes, they have different behaviors. How can you explain that if we are only governed by central dogma, genetic binding based? No, wait, I'm telling you that wait, differential gene expression is one, definitely, no. Even genetically same people, they have different dispositions. These kind of basic interactions between molecules, they have not been explored out of active site. So when light falls on a plant's chloroplast, what happens there is a random ejection of electron that cannot be controlled completely. That is supposed to go through a chaotic process. This kind of chaotic processes are not yet.
No, but even like you were saying yesterday, there are, it doesn't mean that all of those, that all of the ribosomes are going to, bind and they're going to transfer.
Absolutely not.
Some of them, into one cell you may have more, and another cell you may have less. These are not, nobody is saying that this is deterministic.
Absolutely not. There is stochasticity in there. But ultimately, but
Every reaction is stochastic.
yes. But ultimately it goes through that filtering mechanism of determinism at the active site. That is a difference. Now that is a fundamental thing. Now ultimately, one thing gives a particular, now you have n number of nucleotides, they come, they give the exact same stoichiometry, everything based on that particular crucible, that active site. That is determinism.
Even then there are errors.
Yes, there are errors, but then
Holdings and,
now what we are talking about is totally chaotic, molecules mixing and classical chemistry principles governing the outcome.
it's not totally chaotic.
Yeah, it is. There is some kind of.
Like you were saying in the vision, if you are totally chaotic, you will not see anything. You will just see white. You will just see.
It is like pouring water over a mountain. There is some chaos, but then it has a flow. It has a flow. Some of it will stick somewhere, but it will come and get down into the valley. That's what I mean.
So, can I say something?
Yeah, sure. Thank you.
So, when we talk about deterministic and stochastic, so systems, if you don't go into quantum mechanics, if you solely rely on Newtonian physics, then every system is a deterministic system, right? And if it is, systems are always deterministic. It is the approach that is stochastic or deterministic.
Yeah. Definitely.
Stochastic means we don't know much about the system. Right? So there are enough details which we cannot account.
Absolutely.
And that's why it is stochastic.
Yes.
It's a sampling. It's a macroscopic sample.
Absolutely.
Which has a distribution. It's not
Yeah. What I'm saying is, for a semantic reason, just the convenience of using the words to contrast with each other, nothing else. So, with your permission, can I just, the meeting was about the problems, open problems in thermodynamics in biological systems. I think I just made a note of things that is pretty useful, I think. The fundamental problems in dealing with biological systems, you are all physicists, I'm a biologist, and I try to understand things in my own little way. So, energy efficiency and coupling methods are the most fundamental problems, difficult problems to study in biological systems. How in physical systems, you can easily study them, understand the changes and all that, but in biological systems, how exactly an electrical gradient is converted to a chemical bond formation. You cannot really study that. It's a very difficult process. So coupling mechanisms and efficiency, calculation, all that in a complex system is very difficult to understand real thermodynamics in an experimental way. Then how does the system manage the entropy that comes in, and still maintain a working way so far away from equilibrium? That's very difficult to study. And measurement of entropy, first and second law trailing, very difficult. So the classical problems that are currently open are primarily ion pumping. How does ion pump high amount of potassium inside, sodium outside? That people could not explain. Then low.
How is that maintained?
Yeah. So they said that there's Jens who got a Nobel for this sodium potassium pump. 3 sodium outside, 2 potassium inside. Electrogenic ion pumping. Very simple perspective from thermodynamic perspective. Thermodynamics gives the answer to this. I have shown that in the Journal of Cellular Physiology. It's very simple. Potassium is more co-solvated with carboxylates and phosphates in the cell. But the redox potential of potassium and sodium are very different, and that's why we have more potassium inside. So it's thermodynamics that determines, not selective ion pumping, filtering, nothing like that. So then most of the reactions, then fully reversible enzymes is a very false idea that lactate dehydrogenase, for example, when you exert, you have lactic acid accumulated, that has to go to liver. When liver has only 1/5 of the lactate dehydrogenase. So our heart has another isosign. They said that heart does not have lactate accumulation, though it works continuously. That is, they got the idea wrong, because it is not lactate dehydrogenase working reversibly in the liver. It is oxygen-centered reactive species that are present in liver that drive the reaction through a different process. So the idea that all enzyme reactions are reversible is completely wrong, particularly because lactate dehydrogenase has got a free energy change of minus 475 kilojoules per mole. That, you cannot have it like that. It defeats thermodynamics. So things like that, with the involvement of oxygen, the reaction goes through a different pathway and you have it feasible. So thermodynamics is the mother of sciences. It has to be applied, and even in living systems also, you can use thermodynamics, even if it is far away from equilibrium, you can use the basic principles to get the solution. So how can erythrocytes, the red blood cells, they don't have nucleus, they don't have mitochondria, but yet they sustain for 120 days. How do they? No intelligence, no energy. Very simple. Oxygen-centered, oxygen ion. It can dissociate as ion 3 plus superoxide also. Superoxide can bind to attack phosphate, and by the radical pathway it can give ATP. So very simple ways, we have simple chemical reactions explaining the viability that we see outside. So Marcus theory is what we use for explaining electron transfers now. When I was doing a postdoc with Jeff Jones in Washington State University, I was ironically asked that, do you understand this electron transfers of this Nobel recognized idea? He looked up. So now we have a new theory. Yesterday night, thanks to this discussion, I was motivated to look into it, and now we have kind of formulated a mathematical expression that is not exactly Marcus theory, but we have something that can start from thermodynamics and come to kinetics. It's very crazy, but that is what Marcus did, and that is what we can do. Thermodynamics can dictate kinetics. Thank you so much. Thank you.
Thank you.
Yeah, no problem.
Yeah.
Okay, thank you all panelists to give the time and to enlighten us with a nice topic. And thank you also audience. And also as an organizer, I must be sorry, because actually this is a real scenario that we cannot organize lot of audience. Okay.
That is actually sad.
This is sad, but I don't know. Yeah, and that will